Hello, ladies and gentlemen. My name is Habiba Abdelhalim, and I'm a senior research assistant at the Ahmed Lab at the Rutgers Institute for Health, Healthcare, and Aging Policy. Today, I'm going to be presenting our annotated gene disease database and web application for translational research in genomic and precision medicine. Before diving into gene-disease relationship, let us take a look back at the evolutionary field of genomics. The term gene was first introduced through Mendel's experiments in 1909. A few decades later, after identifying the structural model of the DNA, the first DNA was sequenced using Sanger sequencing. Due to the advancements in the genomic field and the application of next-generation sequencing, we are now able to perform single-cell and long-read sequencing. Recent developments in sequencing technology have greatly aided researchers in understanding the alterations that happen in genes. Mutations, which occur when a change happens to the structure of the gene, is now understood to lead to a change in the phenotypic expression of this gene. It is imperative that we focus on the gene-disease relationships to offer a more personalized treatment approach and prevent diseases. In order to emphasize these relationships and understand them better, we have investigated 53 existing gene disease databases. Some of the databases highlighted in our study include, but are not limited to, ClinVar, DrugBank, and GeoDatasets. In the figure shown here, produced from our study, the blue represents the affirmative, while the red signifies the negative. This figure indicates that there's no database in the world currently available that highlights gene-disease relationships, allows the user to input multiple genes at a time, and link disease phenotypes to the ICD codes. This means that after acquiring records from these databases, researchers would still have to go through the time-consuming process of collecting information from other sources and integrating them. More on this study can be accessed through the publications shown in this slide. I invite you to access this publication if you're interested in learning more. There are still difficulties that are faced when trying to connect genomic and clinical data. Currently, there are no clinical genomic databases that are standardized and up-to-date. Additionally, the inconsistencies in gene annotation and the specificity of the disease classification adds another layer of complexity. We need to create tools or databases that can streamline the data integration process of this heterogeneous data. The focus of our study was to collect, explore, and share gene and disease information to support pathology and epidemiology for the implementation of precision medicine. In order to address these concerns, we have created a centralized gene, single nucleotide polymorphism, disease drug database that stores, organizes, and shares data in a structured and searchable manner. We have created a smartphone application called the PROMIS App Suite, or PAS for short, that facilitates data retrieval from this database. The PAS is an integrative application that includes human reference genomes, as well as gene-disease associations. Information on the download instructions and design of the application, as well as the database curation process, are available freely and can be accessed through the following publications on this slide. While the PAS app provides an up-to-date record of gene-disease relationships, as well as drug and disease codes, the information is still not integrated efficiently. This means that users have to navigate between different menus to collect the desired gene-disease code data. Creating one interface where the different types of data is connected allows the user to save time. It would also be used as a tool to diagnose and treat patients accurately and efficiently. So now the question remains, why is it really difficult to integrate gene and clinical data? For starters, clinical data is currently available as standalone. This means that in order to link a gene to a disease and then an ICD code, a researcher would have to access multiple databases. Since there are different types of diseases that arise from one gene mutation and each disease has a unique ICD code, there are many connections that need to be made to reflect this information in one interface. This process can be extremely time-consuming and labor-intensive. In order to begin addressing this issue, we have created and executed the PAS-GDC Code Web Application, or PAS-GDC for short. The gene disease database was manually curated using the 73 actionable genes from the American College of Medical Genetics and Genomics, or ACMG for short. The corresponding ICD-9 and 10 codes were obtained from the World Health Organization. The database was connected to our web application using server-side PHP language, and a user-friendly, searchable interface was adopted to present this data to the user. Our PAS-GDC web application allows the user to search for genes, disease, and ICD codes. It also gives the user an option to search between ICD-9 or 10 codes based on their preference. The website allows the user to export their results in a CSV format. This website is freely available and can be accessed by clicking on the URL shown on this link. In order to search for a specific gene, the user will select the gene checkbox and type in the gene name. For instance, if a user is interested in finding out the disease phenotypes and ICD-10 codes associated with the BRCA1, a gene that codes for tumor suppressor proteins, and mutations in this gene are known to cause breast cancer, a simple search on our website would show that there are 252 ICD-10 codes associated with the breast cancer 1 gene. Users can also search for genes and ICD codes based on a specific disease by checking the disease checkbox. A user wanting to study heart disease, the leading cause of death in the United States, can easily do so. The search shows that 18 different genes are known to be associated with heart disease, and 497 ICD-10 codes are linked to this disease. Similarly, a researcher interested in obtaining the disease phenotypes and gene names associated with an ICD code can select the ICD-9 or 10 database and click on the ICD code checkbox. A simple search of G300 in the ICD-10 database shows that this code is associated with Alzheimer's disease and that there are 2 distinct genes that are linked to this disease phenotype. In conclusion, our PAS-GDC web application offers a freely available, easy-to-use, and up-to-date gene disease code database that can help pharmacists, doctors, and researchers to engage in a more personalized approach to medicine. In the future, we plan on expanding our database to include more than the 73 ACM genes currently available. That concludes my presentation. Thank you so much for your attention.